Hello, this is a primary literature review on the paper about patients with familial adenomatous polyposis, or FAP, harbor colonic biofilms containing tumorigenic bacteria. So, colorectal cancer can be sporadic or hereditary. About 5% of people with colorectal cancer have familial adenomatous polyposis, or FAP, which is a type of hereditary colon cancer caused by a germline mutation in the APC gene. The APC gene is a tumor suppressor gene that regulates beta-catenin and colonic crypts. FAP varies in onset and frequency of polyp formations, even between family members, which indicates that other factors such as the microbiome may be affecting the onset of colorectal cancers. The colonic epithelium is protected from potentially pathogenic bacteria by a dense mucus layer. Atop this mucus layer are microbial communities, which we would refer to as our microbiome. Microbiomes vary from person to person, primarily because of diet, antibiotic use, but also genetics, age, and lifestyle. And the mucus layer limits this interaction from the bacterial layer to the epithelial cell layer to prevent a mucosal inflammatory response. But a lack of a mucus layer may lead to chronic inflammation in the colon. Biofilm formation happens when bacterial quorum secrete extracellular polymers to facilitate attachment and matrix formation. And when the mucus layer is thin, it leaves the body susceptible to biofilm formation, and that can increase inflammation of the epithelium in the colon. Biofilms can cause inflammation because the epithelium layer is not being protected, and sometimes these bacteria causing the biofilms, well, actually regularly, bacteria creating biofilms in our bodies are pathogenic, so they would induce inflammation. The experimental question of the paper is biofilm formation in the mucus layer an early event in the progression of FAP? So first, they performed colonectomies on FAP patients. They used the colon tissues to identify the bacteria that were present in the mucosal layers of the FAP patients. And they used colon biopsies from individuals that don't have FAP as the healthy controls. They did a fluorescent in situ hybridization assay using a probe of panbacterial 16S ribosomal RNA with fluorescence attached, and bound that to cells from polyps and the visibly normal tissues. The probes hybridized via complementary base pairing to the matching DNA in the cell, and it's used to identify bacterial species in mucosal samples from FAP patients and the controls. Samples where the bacterial biofilms were detected had additional screenings performed, where they used additional 16S rRNA probes to identify specific bacteria based on their 16S ribosomal fingerprint. They differ from species to species of bacteria, and they were screened again to observe the major phyla detected from the primary screening. They cultured preserved frozen mucosal tissues of 25 FAP patients and 23 healthy patients to identify the bacteria through FISH. They used 2 polyps and normal tissues per patient when possible. Next, they used a laser capture microdissection, which uses a laser beam and a microscope to isolate specific cells from surrounding tissues. They performed this on the mucosal biofilms of the first FAP patients, and with that, they isolated the bacterial toxins present in the bacteria in the mucosal layers from the biofilms, and they used PCR to identify the bacterial toxins. So they used 2 murine models in this paper to test if the co-colonization of the primary biofilm members, which were ETBF, or Enterococcus, or Bacteroides fragilis, secreting a toxin, and the PKS positive, PKS is another toxin, positive E. coli to identify if the colon tumorigenesis is different between the co-colonization or the monocolonization of just one of these bacteria. So they used one murine model that was given AOM treatment, which is giving it a carcinogen to induce a colorectal cancer phenotype. And there was another murine model that was a APC mutant. They had a truncated version of the APC gene to give them a phenotype much like the FAP patients in humans. These mice were either mono-inoculated or co-inoculated with strains of the identified biofilm members. Then they conducted a histopathological analysis where they used a biopsy to confirm the presence of the disease, the progression, and the physical characteristics of it. They did histopathology scoring to quantify the data from the histopathological analysis, which was used to identify differences in inflammation over time in the mono and co-colonized mice. They used flow cytometry, which is a method of characterizing cells, to characterize immune cells from the mucosal membrane of the mice to determine if the type of inflammation contributed to differences in tumorigenesis in the monocolonized and co-colonized mice. They also did a qPCR to analyze the interleukin-17 expression and test the role of IL-17 in co-colonized AOM mice. Interleukin-17 is a cytokine that links T cell activation to neutrophil activation and is a major marker of the immune response in this paper. Flow cytometry was performed again to analyze the immune cells and the innate and adaptive lymphocyte subsets from the mono and co-colonized mice to test whether the co-colonization of biofilm members affected the early colon immune response and production of interleukin-17. They did an ELISA to profile immunoglobulin A responses, and they did this by using stool from the AOM mice that were collected 4 weeks after colonization. Lastly, they cultured human colon cells that were producing mucin in monolayer so that they could measure the mucus depth on top of the cultured cells. And they performed H2AX immunohistochemistry to visualize cellular components and to be able to measure and observe DNA damage, because H2AX is a histone protein that accumulates in the presence of double strand breaks. So the first figure was the fluorescent in situ hybridization assay that displays the normal tissues stained in blue, and the red stain is the biofilm formation. On the left, you can see the dotted line is the epithelial to mucosal layer transition. On the left is the polyp, which has a very large red layer of bacterial biofilm above the dotted line. And then on the right, you see the paired normal version, which below the dotted line has healthy colonic crypts and much less biofilm formation in red above the dotted line. And then in the middle panel, you can see that E. coli are fluorescing in red and B. fragilis are fluorescing in green when they ran a fish assay with the very specific 16S rRNA markers. And then in the very bottom panel, you can see the histopathology. And on the left, you can see the abnormal polyp formation in the tissue. So the two dominant biofilm members were the E. coli that express colobactin and contain the polyketide synthase PKS genotoxic island. So it's genotoxic, so it induces DNA damage. And in vivo and in vitro studies show that this colobactin induces tumorigenesis in the AOM-treated IL-10 deficient mice. The other major biofilm member is the Bacteroides fragilis, and it's a enterotoxigenic type named ETBF, which is what I'll refer to it in this presentation as. And that is producing the B. fragilis toxin, BFT, which is seen to increase the interleukin 17 levels in the colon. So that first fluorescent in situ hybridization assay is quantified by this table, which is figure 2A in the paper, and it shows us the presence of the PKS positive E. coli and the ETBF on the colon mucosa of FAP patients versus the healthy controls. 70% of the healthy controls had neither type of bacteria colonizing their colon, while 52% of FAP patients had both PKS positive E. coli and ETBF in their colon in this biofilm. So p-values that are less than 0.05 mean that we can reject that there's no significant difference between FAP patients and controls. So all of these p-values in the bottom row of the table are below 0.05, showing that there is a significant difference between the composition of biofilms in FAP patients versus healthy controls. For having neither, 70% of controls had neither, which has a p-value difference between the FAP patients of 0.003, really, really small, means that there's a really big difference there. There's a significantly higher incidence of the colonization of PKS positive E. coli, ETBF, and both present in the FAP patients as well. So figure 3B shows us that the inflammation between healthy controls and the mice colonized with the PKS positive E. coli and ETBF are not significant, which means that inflammation is probably not the mechanism by which they're using to induce tumorigenesis. Figure 4C, the bottom right, shows us that PKS positive E. coli alone had no difference on the mucus depth, but the PKS positive E. coli with the ETBF and just the ETBF had significant decreases to the mucus depth. You can also see that they used Akkermansia muciniphila, which is a known mucin degrading bacteria, as a control in the second column on the graph. So figure 4D is the immunohistochemistry of the distal colonic crypts from the AOM mice that were mono or co-colonized with bacteria. H2AX is a histone protein that appears when double strand breaks occur, meaning that this graph, the higher occurrence in the ETBF and co-colonized mice have a higher rate of DNA damage, and the p-values are below 0.05, so this is a significant difference in DNA damage in the presence of ETBF and the PKS plus E. coli and the ETBF samples. So this suggests that the co-expression of the BFT, which is the Bacteroides fragilis toxin, and the colobactin from the E. coli may be good targets for prevention of colorectal cancer. So the co-colonization with the ETBF and the PKS E. coli promotes carcinogenesis through two mechanisms. There's the mucus degradation, which enables an increased PKS and E. coli adherence, and it induces colonic epithelial DNA damage with the colobactin and Bacteroides fragilis toxin. And the IL-17 induction is promoted by the ETBF with an early augmentation by PKS plus E. coli co-colonization. The mechanisms produce cooperative tumor induction in co-colonized AOM mice, and the constant co-colonization in colon mucosa may contribute to the pathogenesis of familial adenomatous polyposis. It could also be true for those who develop sporadic CRC that the mutation or APC loss occurs in the majority of sporadic colorectal cancers as well. So it's observed that the PKS plus E. coli strain was only cultivatable from the colon lumen in the absence of the ETBF colonization in the mouse model, which is consistent with observations that PKS positive E. coli and ETBF co-colonize the FAP colon biofilms. And data suggests that there's a link between early tumor growth and tumorigenic bacteria, meaning that in the future, we can analyze the co-expression of the Bacteroides fragilis toxin and colobactin in the efforts to prevent colorectal cancers and in screening for them. Thank you.